Recombinant bacterial antigen, edible vaccine case, presented by Ali Voronova. Diseases. According to World Health Organization, infectious diseases caused by the pathogenic microorganisms such as bacteria, viruses, parasites, and fungi. The diseases can be spread directly or indirectly from one person to another, causing epidemics, and based on World Health Organization statistics, thousands and thousands of deaths worldwide every year. The worst Ebola outbreak in history has killed nearly 5,000 people and infected more than 10,000 in West Africa. The high mortality risks, according to the World Health Organization, occurs from such diseases as tuberculosis, HIV, diarrhea, childhood cluster diseases, and hepatitis B. The last one kills nearly 800,000 individuals every year worldwide. But the prevention of all these deaths exists and can be done through immunization. As language states, vaccine has accomplished nearly miracles in the fight against infectious diseases. However, according to the same author, as well as World Health Organization, 20% of infants globally missed by 6 vaccines against diphtheria, whooping cough, polio, measles, tetanus, tuberculosis, account for about 2 million unnecessary deaths each year, especially in most remote and impoverished parts of the globe. The situation is worrisome not only for the places that lack health care, but for the entire world. The question was how to get more efficient in a way to produce vaccines that could reach everyone who cannot afford them due to the cost of simply non-existence in certain regions of undeveloped countries. In early 1990s, this question was brought up by the scientist Charles Arnsten, Texas University. He's a major contributor in the field of plant molecular biology and protein engineering. Plant biologists had already devised ways of introducing selected genes, the blueprints of proteins, into plants and inducing the altered or transgenic plants to manufacture the encoded proteins. Thus, Professor Arntzen proposed the idea of edible vaccines. As a group of scientists states, introduced as a concept about a decade ago, edible vaccines has become a reality today. Multiple sources postulate that edible vaccines can be cost-effective, easy to administer, easy to store, easy to deliver for the poor developing countries, could be grown locally, standard growing methods of a given region, and can be possibly more efficient vaccines than standard injections. The idea is simple. It involves introduction of selected desired genes into plants and then inducing these altered plants to manufacture the encoded proteins. Well, how to make an edible vaccine? One way of generating edible vaccine relies on the bacterium, agrobacterium, to deliver into plant cells the genetic blueprints for viral or bacterial antigens, proteins that elicit a target immune response in the recipient. Well, this diagram, as you see, can show how it exactly works. For example, you cut a leaf from the plant, you expose leaf to bacteria carrying an antigen and an antibiotic-resistant gene. Allow bacteria to deliver the genes into the leaf cells. Then you expose leaf to the antibiotic to kill cells that lack the new genes. Wait for surviving, gene-altered cells to multiply and form the callus. Then allow callus to sprout, shoot, and root. Put in soil. Within 3 months, the plantlets will grow into plants, bearing the antigens, vaccine potatoes. Classic vaccines pose a small but troubling risk that the vaccine microorganism will somehow spring back to life, causing the diseases they were meant to fight. For that reason, vaccine makers today favor so-called subunit preparation composed primarily of antigenic proteins divorced from pathogen genes. Food vaccines are like subunits preparations in that way. They are engineered to contain antigens, but bear no genes that would enable whole pathogens to form. Edible vaccines come into contact with the lining of the digestive tract, and in theory then they would activate both mucosal and systemic immunity. The dual effect should in turn help improve protection against many dangerous microorganisms. Professor Arnsten and his collaborators from Cornell University and Loma Linda University developed tomatoes and potato plants that can synthesize antigen from the Norwalk viruses, E. coli, cholera, and hepatitis B virus. Researchers explain that antigens delivered in plant food survive the trip through the stomach well enough to reach and activate the immune system. And again, here we can apply the statement that we learned through the course that structure dictates functions. The tough outer wall of plant cells apparently serves as a temporary armor for the antigens, keeping them relatively safe from gastric secretion. When the wall finally begins to break up in the intestines, the cells gradually release their antigenetic cargo. The scientist Hilary Kaprovsky from Thomas Jefferson University, for example, fed transgenic lettuce carrying hepatitis B antigen to 3 volunteers, and out of 3 volunteers, 2 of the subjects display a good systemic response. It's not a substantial research, but it's very interesting and very promising. Edible vaccines are a promising form of immunization that requires more financial, scientific, and public support in order to reach millions of infants and eliminate the risk of epidemics outbreak and unnecessary deaths worldwide. Building this project was very interesting and informative for me. Such a great technologies as gene expressions and recombinant bacterial antigen introduction in genetically modified products have a great and very promising future, I believe. What I've learned through this project. I've learned that plant structure can be very powerful and suitable for vaccines distribution. The technologies is on the rise and to be fully developed and incorporated in our health system. The benefits are enormous and vitally important for the public. However, any innovative technologies or research leaves some questions that needs more detailed investigation to be answered. In one of the articles, the researcher William Langridge that I stated previously proposed a very interesting question, the answer to which I would like to investigate more. For example, if such vaccines exist and people start consuming them, would food vaccines ingested by mothers indirectly vaccinate their babies, either through placenta or through milk? Additionally, I'm very interested in the idea that food, thanks to new technologies, might become more nutritious. I would like to investigate the possibilities of creating such a food and their benefits in fighting malnutrition, especially in undeveloped countries. Thank you so much for your attention. And if you have any questions, there's a list of references I used for my project, as well as you can contact me anytime. You know my email, and please don't hesitate. I would love to answer any of your questions. Thanks once again. Goodbye.